Welcome back to part 2 of week 9. We are now into DNA replication. So we're going to start with, of course, the little review of what we just covered. We have DNA as this double helix with 2 strands of nucleotides, a nucleotide having that nitrogenous base, the sugar, and a phosphate group. And now we say these are anti-parallel, or there's directionality to DNA because how that strand is oriented. You can have the 3' end sticking out, so that's the 1, 2, 3rd carbon is going to be at one end, which means the other end will always have the 5', the 1, 2, 3, 4, 5th carbon, which is attached to a phosphate group. So if you have the 3' end here, the opposite side is going to have the 3' end. So 5, 3, 5, 3. And it's the hydrogen bonds connecting those nitrogenous bases. As you should have seen in the video that you watched for the lab for this week, talked about Watson and Crick and how just seeing the structure of DNA with that sugar phosphate backbone and those nitrogenous bases forming those base pairs sort of tells us, oh, the structure of DNA explains how it can use itself as a template to make more of it. So we're going to now cover the process, how one section of DNA can use itself as a template to replicate. So we have DNA replication. So at the very basic level, this is what you need to understand. The DNA is a double helix that is going to get separated, and we're going to learn about all those proteins. And each strand of DNA is going to serve as a template by knowing that A's are only going to bind with T's and C's will only bind with G's. So that if you separate one strand, you can make the corresponding strand just based on those base pairing rules. Of course, you need to know it at a greater level than this, but first step, this slide should make perfect sense to you. So this idea that the strand itself serves as a template and that template then is made up of an old strand and a new strand is called semi-conservative replication. There are other ways this physically could be done. And in fact, in the worksheet you did, you sort of looked at what would be the results if DNA replication worked differently. But we can see from the data, it is in fact a semi-conservative model. So now we're going to dive into how this semi-conservative replication actually works. You can watch this nice overview video. And we're going to start by just acknowledging the difference between prokaryotic and eukaryotic replication, where in those bacteria cells, they of course have a chromosome. Some people in an earlier assignment were explaining that they don't, but bacteria absolutely do have a chromosome. They just have one, and it's a nice circle. But it does need to get replicated. And in that, there's an origin of replication where that is going to start. And it's going to unwind, and there's going to be new strands copied going in both directions, both for, in this case, the top strand and the bottom strand. So both strands get copied to make 2 complete exact same copies, and those are both going to go into the 2 daughter cells, where one of the strands is the old strand and one of the strands is the new strand. In our eukaryotic cells, it's basically the same thing. There's an origin of replication, but in fact, in our much longer, much more plentiful DNA, there's actually multiple bubbles and multiple origins of replication that come together as that replication fork continues to move closer and closer to each other. So there's the origin of replication, there's the replication fork, which is where the double helix is opening up. There's the old strands and the new strands. So I recommend watching this video. I'm also going to show this video that I'm actually going to play the whole thing right now, and it's going to be the best overview that's sort of setting the scene for what we're going to talk about, where we are going to need to know all of the proteins.
Before a cell can divide in two, it must first duplicate all of its DNA so that it can pass an exact copy to each daughter cell. DNA replication is carried out by a molecular machine called the replisome that pulls apart the double helix and makes an exact copy of each strand. The DNA to be copied enters the production line from the left. The blue ring-shaped molecule is the enzyme helicase, which unwinds a double helix into two strands. The leading strand is copied continuously. The lagging strand runs in the opposite direction with a more complicated copying mechanism. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
So that was the overview. Now we're going to start to dive deep, and I'm going to first do an overview again in a picture form and then go into the more depth that you will need to know. I'll also comment that you are going to need to be making a video similar to this, describing the process of DNA replication. So we have your origin of replication will be way down here, but we have our replication fork right here, and we have these enzymes, and this enzyme is opening up the double helix. We can also see there's two different strands going on here. We see in this case, we're going to hear this is the leading strand where there is a protein called polymerase, which is going to be adding the nucleotides, and it's going to go continuously in the direction that the helicase is opening up. Now there's this other strand over here that we're going to learn is called the lagging strand, and this is going to be made in segments, but it still has that DNA polymerase adding the nucleotide. And that protein, DNA polymerase, is kind of finicky. It can't just add on nucleotides just anywhere. It can't start someplace and just begin. It needs to actually have something it's adding to. It needs that free 3' end to add to. So there is another enzyme called primase, which is going to deposit a primer or a few nucleotides. And that is what DNA polymerase will work off of. So we see in the lagging strand, like we did in the video, that there's multiple sections that get pulled out for this lagging strand. Ultimately, they are linked together by a protein called DNA ligase, which is ligating or acting like tape to fill in and connect that 3' carbon from one sugar to the phosphate group attached to the 5' carbon of another. So now we're going to get into a bit more depth, where again, you have the replication fork, and it's being opened up by helicase. There are also additional proteins called single-strand binding proteins, which do exactly as their name suggests. They bind single-stranded DNA. So they are binding this DNA to make sure that after helicase has opened it up, it doesn't snap back together. We can also see here you have this leading strand where DNA polymerase is just constantly adding nucleotides to this free 3' end. And it is this property of DNA polymerase, which can only add nucleotides to the 3' end, which make for a leading strand and a lagging strand. Because DNA can only be added going in this way, so that it's only the polymerase from this 3' to 5' end that can continually go. For it is on this side, the lagging strand, that polymerase is going to keep adding nucleotides, but only in this direction. So it's going to be going that way. We can see here, though, that we have primase. Primase is the enzyme that is depositing the nucleotides that make up the primer. Now, other important things to know about the primer are that the primer is made up of RNA nucleotides. RNA nucleotides. So primase is depositing RNA that is going to be the facilitator, is going to be the helper for DNA polymerase to add nucleotides to. So we can bring this out even more and see exactly everything that's going on. Again, we have the replication fork that's being opened by helicase and those single-stranded DNA binding proteins keeping it open. We have DNA polymerase 3 starting at the origin of replication for the leading strand and just continuously going, adding nucleotides. But on this other lagging strand that's being pulled out in snippets, we have these fragments. We have these fragments where essentially that DNA polymerase has to be going the opposite direction. because it's always going to be adding nucleotides to the 3' end. So you're always adding to that 3' end. So DNA is always made from the 5' end to the 3' end. This is why it was so important to understand which carbon was which and how those DNA strands are running anti-parallel. So we have primase making those primers made up of RNA nucleotides. We have DNA polymerase adding bases to those nucleotides. And then we have ligase, which is going to seal those bonds between the different fragments. Those different fragments are called Okazaki fragments. Now, I mentioned that primase is adding RNA nucleotides. There is an additional different type of DNA polymerase, which is going to remove those RNA nucleotides and replace them with DNA nucleotides. So that is called DNA polymerase 1s. So you need to know all the different proteins and what's going on for how DNA is replicated. I have some key terms here. And it's also important to talk about the fact that DNA polymerase isn't always perfect. DNA polymerase can make mistakes. In fact, in the next few slides, I'll show you some things that causes DNA mistakes. But now DNA polymerase also has what we call proofreading capacity, where it can actually see that it's made a mistake, remove that nucleotide, and add right in the correct one. So this actually involves different proteins, and this is really important for making sure that we aren't causing changes in our DNA. Because a change in our DNA we call a mutation, and we'll learn more about the fact that mutations can cause differences in our proteins in the next few chapters. But uncorrected errors can cause serious problems or no problems at all. And we're going to talk later again about the difference between our gametes, which we're going to learn about are our sex cells, so our sperm and our eggs. So a mutation in a gamete would be passed down to generation. A mutation in a somatic cell we're going to learn about, that's our skin cells and our liver cells and basically everything that's not a gamete. So everything that makes up you that's not going to make up your babies. So a mutation to a somatic cell is going to affect you. This mutation in a gamete is going to affect your offspring. So how do some of these mutations arise? So remembering back to that energy spectrum radiating from the sun, there's ionizing radiation. So these have more energy. So this is X-rays and most of the UV light. And we're going to see that ionizing radiation literally steals ions and forms breaks. So there's literally breaks in your DNA. So DNA polymerase makes a mistake because there's a literal hole. There's also non-ionizing radiation. So this is a little less energy, but it still causes significant problems. So there's something called thiamine dimers, where two thiamines next to each other, instead of forming their normal base pairs with those adenines, form this structure. And basically anything that makes DNA kink like this, it forms this kind of weird bump, makes that polymerase that's charging through and going super fast, as you saw in that video, real time, it can make a mistake. And thiamine dimers are frequently caused by exposure to UV light. So what happens when you get too much sun is you can form these thiamine dimers in your skin cells, which then polymerase can make a mistake and can cause a mutation, and this sometimes leads to skin cancer. Other mutations are caused by things that you also think of as causing cancer. How do they cause cancer? They cause a structure of the DNA to change so that polymerase screws up and adds the wrong nucleotide. Things like smoking tobacco can form methyl groups, so carbon with 3 hydrogens, and that forms this like just kink that makes that polymerase stutter and can screw up. The good news is for the most part, our bodies are able to change those mutations and fix them, but it's also the basis for evolution and having those changes. And we won't get to talk too much about it, but things like viruses actually want to make changes, want to have that variation, and that's one of the mechanisms by having polymerases that make a lot of mistakes. So viruses have polymerases that intentionally kind of make mistakes so that they can change. Lastly, I want to check your understanding. So I post these, and this is not, I post the answers in the PowerPoint, but I have them here in the video. Number one, you want to represent, what are those dots? Those dots are a hydrogen bond. You want to label the 5' and 3' ends. Well, you got to see 1, 2, 3. So if this is my third carbon, then this side is going to be my 3' end. And right here, 1, 2, 3, 4, 5, this is my 5' end because it's where the phosphate group is. You always know it's your 5' end because that's your phosphate. And if this is your 5' end, even without counting, I know this is my 3' end. If I know this is my 3' end, I know that down here is going to be 5'. So draw an arrow pointing to a purine. So these are any of the adenines or guanines, any of these two-ringed structures. If the origin of replication was at the top right here, which strand would be the template for the leading strand? So we have to remember the leading strand is always going to be going from the 5' to 3' direction itself. It's going to keep getting nucleotides added to it. So what's going to be the template for that 5' to 3'? That's going to be the opposite, the 3' to 5' then. Example then, if this is the leading strand template, this is going to be the lagging strand template. And then lastly, to draw a box around one pair of bases, so one base pair, adenine and thymine right here. I could have also drawn it here or here or here. Another important check of your understanding. So here we have this replication bubble. You have the origin of replication here. I have the 5' end here. Can you label all the other ends? Well, if this is 5', then this is going to be 3'. If this is 3', this is going to be 5'. If this is 5' here, this is 3' over here. If this is 3', this is 5'. You see where I'm going with this? 5', 3'. So you're always going to have those opposites. So if here's the origin of replication, where would helicase be? Helicase is going to be at these replication forks, opening up that double strand. Can you draw the other strands and label them? So here we're going from the 5' to 3'. This is going to be a leading strand, which makes this side going to be that lagging strand going in bits. Equally, down here, still have the origin of replication, everything going here from that 5' to 3', that's going to be leading. Everything going this way is going to be that lagging strand. There's 4 RNA nucleotides, 1, 2, 3, 4, that are making up this primer that were deposited by primase. So what's the sequence of those? That's going to be UUGG. So uracil, uracil, guanine, guanine, because adenine binds with thymine and DNA, but uracil and RNA. And C's always go with G's. What was the name of the enzyme that made that RNA piece? Primase. And what's the name of the enzyme that's making the new DNA strand? That's DNA polymerase 3, which is adding nucleotides that 3' end. And with that, we've covered the material for week 9. But if you don't understand how to answer questions like this, you need to go back and keep reviewing. Good luck.